Dr. Holder is an associate professor of pediatrics and neurology at Baylor College of Medicine in Houston. He earned his MD, PhD at the University of Texas Southwestern. His PhD focused on genetics and development, and he did his residency in pediatrics and neurology at Baylor College of Medicine and postdoctoral fellowship in neurogenetics at the University of California, San Francisco. Dr. Holder is currently a neurologist at the Bluebird Neurology Clinic at Texas Children's and main investigator at the Holder Lab at the Jan Duncan Pediatric Neurological Research Institute in Houston. The Holder Lab's primary research is in genetic and neurobiological basis of neurodevelopmental and neuropsychiatric disorders. Their goal is to discover individualized therapies for neurodevelopmental disorders. SYNGAP is one of those main areas of study, and using cell-based screens to mouse models of disease to neurons derived from induced pluripotent stem cells, or iPSCs, of SYNGAP1 patients, they try to find genetic modifiers of the protein stability to identify therapeutic entry points for SYNGAP1. His lab has also a focus on finding biomarkers and deep phenotyping, investigating particular areas like sensory, sleep, and walking patterns of SYNGAP1. Their ultimate goal is to translate the findings from the lab back to the clinic to improve the quality of life of children with SYNGAP1. Dr. Holder has participated in several paramount publications in SYNGAP and is considered one of the clinicians and researchers with deep knowledge of the disease. He is extremely compassionate, enthusiastic, and kind when seeing his SYNGAP patients in his neurology clinic. We are truly grateful for the work that he has done and his interest in SYNGAP1. A recorded version of the webinar will be available on the SRF website under webinars. By the end of this presentation, you will have the opportunity to get the answer to your questions. We'd love to hear from you, so please write your questions in the Q&A below. And for those of you just joining us, welcome. And again, our speaker today is Dr. Jimmy Holder, and his presentation is Further Lessons from Prospectively Deep Phenotyping Individuals with SYNGAP1-DEE. It's now my pleasure to turn things over to Dr. Holder.
Well, thank you so much for that introduction. Good morning, everyone. The introduction was more than I deserved, but I appreciate it. Thank you so much. Today I'm going to really focus mostly on phenotypes or symptoms associated with patients with mutations in SYNGAP1, which is an area that I've been interested in, I guess now for over 5 years. So today I'm really going to focus on two newer studies, one that was just published in December of last year, and the second that we're still analyzing the data. We've collected all the data. We're still working on analyzing it and hope to submit that for publication this year. But I'm going to give you kind of a sneak peek at some of that data. And then at the end, I'm going to kind of wrap up by just really quickly going through some of the previously published data, some things that I've presented here before, but there might be some new families that are joining us today that haven't seen some of that information. And then, of course, I'll be happy to take your questions. So a couple of disclosures. I will talk about some off-label use of medications very briefly, and I'm a site PI for a number of clinical trials. So it's now been over a decade since mutations in the SYNGAP1 gene were first identified in 3 little girls with intellectual disability. This work was published from Jacques Michaud in Canada. And what he found were 3 de novo loss-of-function mutations in the SYNGAP1 gene associated with, again, intellectual disability. Since that really landmark paper published in 2009, there were rapidly a number of additional papers broadening the phenotype associated with SYNGAP1 mutations to include epilepsy, autism, and of course, intellectual disability as being hallmark phenotypes associated with mutations in this gene. And then in 2020, what I really consider a landmark paper published in Cell came out, in which a group of labs collaborated together to try to understand the genetic landscape associated with autism. To do this, they sequenced over 10,000 individuals who had clinical diagnosis of autism and evaluated for de novo loss of function mutations in genes that could be causative. And what they identified were really the largest number of genes that had ever been associated with autism. But what's most important for this group is that SYNGAP1 came out on top as one of the most prevalently mutated genes, monogenic forms of autism. Now, mutations in SYNGAP1 are still relatively rare in this group. They found something around 16 or 18 de novo loss of function mutations. But among all of these really rare causes, monogenic causes of autism, SYNGAP is one of the most prevalent. So I'm just briefly going to talk about what SYNGAP1 does. So SYNGAP1, the gene, encodes SYNGAP1, the protein. And the very first function that was associated with the SYNGAP1 protein was at the synapse, at this point of communication between neurons. This work was pioneered by Rick Huganier and Mary Kennedy. And what they found over a number of publications in a number of years, them as well as others, is that SYNGAP works at the synapse, and specifically within the postsynaptic density, within the part of the synapse that receives communication from other neurons, to regulate other proteins. And in particular, it acts as an inhibitor for a number of small G proteins. Among these, one of the most important is the RAS protein. SYNGAP inhibiting RAS then leads to less activation of ERK by RAS. And that in turn leads to less insertion of AMPA receptors. These are glutamate receptors at the synapse. So what does all of that mean? What that means is that if there are fewer receptors at the synapse, then when there is excitation of a neuron onto another neuron, there is less, it requires more activity for that postsynaptic neuron to become active, to fire. In contrast, when there is a reduced amount of SYNGAP, this leads to overactive RAS, as well as other small G proteins, overactive ERK, and then increased AMPA receptor insertion within the postsynaptic membrane. And that makes it just easier for these postsynaptic neurons to fire. And so the result overall, we think, is some of the behavior issues that we see both in children with mutations in SYNGAP, as well as in mice that have been created in other animal models, as well as this increased excitability probably is the basis for the hyperexcitability of neurons and potentially of seizures. Now, more recent work, and I won't dive into this because this is, you've had other investigators that have talked about this, but more recent work has shown that SYNGAP actually has other functions, including in neurogenesis, as well as in early neuronal differentiation, pioneered by Gavin Rumbaugh and others. So I just wanted to give you kind of an overview of the function of SYNGAP at a molecular level. I really won't discuss this in any more detail. Like I said, the focus of this talk is more on the clinical symptoms and phenotypes associated with mutations in SYNGAP. So what I'll talk about today is our prospective work to understand the symptoms and phenotypes associated with SYNGAP1 mutations, which has been going on now for about 5 years, and has relied heavily on you, the families, to participate, to help us gain this better understanding. So I really appreciate all of your time and effort in that. I know how difficult it is to find the time, even for some of our short studies that are just 15 or 20 minutes long. So again, I really appreciate that. And then at the end, very briefly, I'll touch a little bit on the heterogeneity of clinical phenotypes associated with SYNGAP1 mutations. So a number of years ago, we published this figure demonstrating the genetic landscape of SYNGAP1 mutations. This is certainly outdated at this point. I'm not sure a single figure can include all of the different mutations now that have been identified. But I think this is still useful in that it demonstrates that mutations really can occur anywhere in the SYNGAP1 gene, almost. So we most commonly find loss of function mutations or missense mutations that cause neurodevelopmental disorder in sort of the middle exons, basically between like exons 2 or 3, most commonly 3 out to 18. The SYNGAP1 gene has 19 total exons, and we very infrequently or hardly ever find mutations that cause sort of the classic phenotype associated with SYNGAP1 mutations in exons 1 or 2 or exons 19. And this is probably for a number of reasons. There's a lot of what's called alternative splicing or alternative start sites. What this all means is that although SYNGAP1 is a single gene, it can produce several different types of proteins. And so mutations in the gene that affect all of the different types of proteins are probably more deleterious than mutations that only affect one or two different forms of the SYNGAP1 protein, but leave the other forms of the SYNGAP1 protein intact. So that's probably at least part of the reason why we most commonly find these mutations sort of in this middle part of the SYNGAP1 gene. So as I said, I'm going to focus really on two studies that we have completed. One that's already been published that focuses on socialization behaviors, social behaviors in children and in children with SYNGAP1 mutations. And then the second is some further work that we've done with regards to sleep in children with SYNGAP1 mutations. So really the most comprehensive paper describing the symptoms and phenotypes associated with SYNGAP1 mutations came out now a number of years ago in neurology from Ingrid Scheffer's group in Australia. And she and her colleagues used sort of a standardized questionnaire to talk to individuals really around the world who are caring for children and adults with SYNGAP1 mutations to really dive into what are the phenotypes, what are the symptoms that you guys see. And so she published really a very nice paper describing a number of the phenotypes that are seen and the prevalence of those. Shown here is just one table with some of that data demonstrating that nearly all, somewhere around 90 to 95%, of individuals with mutations in the SYNGAP1 gene have intellectual disability. Of course, nearly all kids with mutations in this gene, or probably around 90%, have epilepsy. But what I really want to focus on is this bottom row here, where she reported that about half of the kids that they, for the families that they interviewed, had a clinical diagnosis, autism or autism spectrum disorder. So, we were interested in this diagnosis of autism and more broadly in social behavior abnormalities in children with SYNGAP1 mutations because this work, while really wonderful and really laid out, what the phenotypes are that we commonly see with SYNGAP1, did rely on, guardian recall or a clinical diagnosis of all of these phenotypes. And so we wanted to look a little bit more quantitatively and more prospectively and in more detail at socialization defects in kids with SYNGAP1 mutations. And so to do this, we used a tool called the Social Responsiveness Scale, second edition, or SRS2. This is a standardized tool that's been used repeatedly in a number of populations, including individuals with idiopathic autism, as well as some other neurodevelopmental neurogenetic disorders, such as Fragile X syndrome, as well as Phelan-McDermid syndrome, which I'll come back to in a minute. It's a relatively simple tool in that it's just a parent questionnaire. And so we recruited individuals both from my clinic here in Houston, as well as really from around the country. Again, a big thank you to all of the caregivers that took time out to talk to our research coordinators and answer our questions. And so I mentioned that this tool has been used in a number of neurodevelopmental disorders, and one of those where it's already been used before is in something called Phelan-McDermid syndrome. And as you'll see in the next few slides, in my group, we very frequently have compared kids or individuals with mutations in the SYNGAP1 gene with individuals with Phelan-McDermid syndrome, which is due to a mutation in this gene called SHANK3. So why do we do this? Well, part of the reason we do this is the SYNGAP1 and SHANK3 function in a similar area of the neuron, of the principal brain cells, and that is in the postsynaptic density. So I've already talked quite a bit about SYNGAP1 and its function in the postsynaptic density, SHANK3 has somewhat of a similar function in that it works both as a scaffolding protein in the postsynaptic density as well as potentially a signaling molecule. And in fact, SHANK3 and SYNGAP1 is shown over on the right here, are hubs in protein interaction networks, and actually either directly or indirectly physically interact with each other. Moreover, sort of looking at in a broad sense, kids with Phelan-McDermid syndrome and mutations in SYNGAP1 have similar phenotypes in that they broadly have, almost all have intellectual disability, about half have autism, and many have epilepsy. So we thought it would be good to compare these two, what we call developmental synaptopathies, two genes that encode proteins that are really critical for how neurons communicate with each other. So for this SRS2 study, to show you the demographics here, we were fortunate enough to recruit overall over 100 families to participate in our study. You can see here on the left, we had about an even number of males and females. If you look over on the right, the age of the three groups that we looked at, kids with SYNGAP1 mutations, kids with Phelan-McDermid syndrome, as well as healthy controls who were siblings of kids either with SYNGAP1 mutations or Phelan-McDermid syndrome. The ages were similar among the three groups. The biological sex was similar among the three groups. There was no statistical difference, although I will say we ended up with a few more kids with a few more girls with SYNGAP1 mutations than boys. But overall, we had a pretty even number of kids of both males and females, and a similar age distribution. So what did the data show? So if you focus first over here on the far left, looking at the total score of kids either with SYNGAP1 mutations, Phelan-McDermid syndrome, compared to healthy controls, we see that kids with both of these neurodevelopmental disorders had significant social impairments compared to healthy controls. And I'll show a little bit more detail about the breakdown of kids with SYNGAP1 and Phelan-McDermid syndrome in terms of who scored in the normal range and the severity of score that they had. But looking kind of broadly, not surprisingly, these kids had significant social impairments. This instrument can also give us some information about different domains of social interactions. And that's shown in the figures to the right here. We can look at social awareness, social cognition, social communication, et cetera. And what we found from all of these is that there are significant impairments in all of these domains of socialization in kids with SYNGAP1 mutations compared to healthy controls. And moreover, statistically, there was really no difference between the kids with SYNGAP1 mutations and the kids with Phelan-McDermid syndrome. So again, looking at a bit more detail here on the lower right side, when we break down the scores based on what's kind of standardly used in the field for scores in the SRS2 for normal range versus those affected, what we found for SYNGAP1 is that only about 10% or so, a little bit more than 10% of these kids scored within the normal limits. And I'm going to talk a bit more detail about that in a slide or two. Whereas about over 80% of the kids we found had definite deficits in socialization. And you can see that the majority of those kids had severe deficits. Slightly different in the Phelan-McDermid syndrome group that we looked at, as well as what's been published before, where we saw no kids in our group with scores within the normal limits on this SRS2 scale. And then again, the vast majority of these kids with Phelan-McDermid syndrome had severe deficits in socialization. And again, all of this is very much in contrast to the healthy controls. So looking back at Ingrid Scheffer's paper in neurology, again, I mentioned that about half or so of the kids with SYNGAP1 mutations in her group that she recruited had a clinical diagnosis of autism spectrum disorder. So why is there this discrepancy? We see over 80% of the kids in our group with social deficits that have SYNGAP1 mutations, whereas Ingrid's group found about 50% are diagnosed with autism. Well, I think there are potentially a number of reasons. One, as I told you earlier, her work was retrospective, so it relied on a physician at some point recognizing that a child potentially had autism and either making a clinical diagnosis just based on interview and an exam, or based on some of the standardized instruments that are used clinically. So if a child just happened to not have seen a clinician that made that diagnosis, that could underestimate the prevalence of autism. The other reason is that autism really requires a number of discrete elements in order for that diagnosis to be made. Whereas what we're looking at is really socialization kind of broadly, not really looking at things like expressive or receptive language in great detail with this instrument. So it's possible that some kids with definite social deficits that we identified through the SRS2 instrument just don't meet all of the criteria for autism. In any case, this gives us a better idea that kids with SYNGAP1 mutations, that the vast majority do in fact have social deficits. We also looked at the data in a little bit of a different way, and that is using something called the short form of the SRS2. So this basically involves excluding some of the questions in the full form, which was administered to everyone. And the reason we exclude these questions is that through previous studies, it's been found that individuals with intellectual disability, that some of the questions are really hard for parents to answer. They're really just not applicable. And some of you that participated in our study may have found that. And so a previous study published a number of years ago evaluated, well, if we exclude those questions that are most commonly can't be answered by families caring for individuals with intellectual disability, can we get a better idea of what's going on? And part of the reason of this is if a certain number of questions are excluded, we have to end up throwing out the data altogether because there just isn't enough data to really do the analysis. And what we found when we did this is that, in fact, we had a number of individuals with SYNGAP1 mutations, I think it was around 8 or 9, that we had to exclude from the analysis using the full form, that we could then, most of them, include them again when we use the short form. And so overall, for this, what we found was very similar, which is that kids with SYNGAP1 mutations, as well as kids with Phelan-McDermid syndrome, have significant social deficits using the short form as well as the long form. I think more kind of to the point, and thinking about this tool as potentially something that could be used as an endpoint for future clinical trials, probably the short form would be a better tool overall for our kids with SYNGAP1 mutations. So next, we were interested, and I'll be honest, this actually came up because when we submitted this paper, a reviewer asked us this question. Is there any change, or is there any difference in those individuals who are a bit older in their SRS2 scores compared to younger individuals? And my first inclination was, well, probably not. There's probably not going to be much of a difference. And when we looked at this in a bit more detail in the literature, this one paper came out that we took a look at, which showed actually in idiopathic autism, so in all comers, all children, individuals you can find with autism that you administer this SRS2 instrument to, you actually have a reduction in their score. So their autism symptoms potentially seem to get better as they get older. And there can be a number of reasons for this. One obvious one is that many of these individuals may have had therapies throughout the years and developed mechanisms to cope with some of the symptoms that they have associated with autism. So their scores potentially improve over time. So we looked at scores in our SYNGAP1 kids, as well as Phelan-McDermid syndrome kids and healthy controls, and correlated those with age at the time that we administered this instrument. And actually, we found something really surprising, which is that in general, the total score on the SRS2 seems to get quite a bit worse as kids and older kids with SYNGAP1 and adults compared to the younger kids with SYNGAP1 mutations. And this is in really stark contrast to the kids with Phelan-McDermid syndrome, where we saw absolutely no difference at different age groups, no correlation between total score and age. And this is both, as you can see, on the full SRS2 form as well as on the short form. And then, of course, healthy controls also, there's no correlation between age and total score on the SRS2. So this was really surprising to us. And, I at this point, I have some hypotheses about why that might be, but I'm not really going to share those because I have no data yet to really address this. But something that I think we and others really need to look at. I'll just point out that if you recall from a few slides ago, I said that 4 kids scored within the normal range on the with SYNGAP1 mutation, scored in the normal range on the SRS2 instrument. And those kids are all very young. So if you look at this upper left graph, you can see those kids are there on the far left at the younger ages. All right, so I'm going to change gears now and move from talking about socialization in kids with SYNGAP1 mutations to talking about sleep. So again, in this really landmark paper from Ingrid Sheffer's group, she looked again at a number of phenotypes associated with SYNGAP1 mutations, and one of the ones that, of course, she looked at were sleep problems. And what she found in this paper was that about 60% or so of individuals that for families that they had interviewed, reported some sort of sleep problem. And this could be, a lot of different things. This could be difficulty initiating sleep, difficulty in maintaining sleep, anxiety associated with sleep, etc. So all different, types of issues with sleep. She found about 60% had issues. So how can we, in a more sort of prospective, quantitative way, address these sleep issues in kids with SYNGAP1 mutations? So I'm just going to outline here a few approaches that one could potentially take. And this includes really what I would say are 3 general approaches. One is using sleep questionnaires. So the prime example of that is the Childhood Sleep Habits Questionnaire. And I'll talk about some data that we and some of my collaborators, Connie Smith-Hicks and Andrew Stanfield, that we published together using this instrument, this questionnaire instrument, to investigate sleep in kids with SYNGAP1 mutations. Some of the reasons to use this, some of the pros for this, is that it's really pretty easy to administer. Those of you that participated with one of our groups, you'll recall it just takes about 15 or 20 minutes to answer this questionnaire, which can be done either in person, can be done just by the parent. You can, we opted to have a research coordinator call all of our participant families or in person to administer it. And it's inexpensive, so it's relatively easy to administer to a large number of individuals. Some of the cons, it does rely on caregiver recall. So, there can be potentially at least some bias there associated with that. Second approach is actigraphy. An example of this is something called ActiWatch, and I'll show you a little bit more detail about this in a minute. Again, a number of you have probably participated with us on this project, so again, really appreciate that. And I will be showing you some data that we've collected from this. Moderately easy to administer. You can get objective data from this. Some of the cons, it does require the participant to tolerate the device, which is basically looks like a smartwatch, and turned out to be a significant problem in our kids with SYNGAP1. As I'll just really briefly mention, these kids have significant sensory processing issues and significant issues with sensory tolerance, and so that ended up leading to a number of kids having to be excluded because they basically just could not wear the watch. And then it's quite a bit longer to collect data for this. Instead of 15 to 20 minutes, this usually requires 1 to 2 weeks of continuous data collection. Although for those kids that tolerate it, you just put the watch on and then let them go about their normal daily activities. then the final approach, which is probably the most quantitative, most objective of all, and you get the most data from, is polysomnography or sleep studies. If any of your kids have had these, you know, this involves usually going into a sleep lab, sort of this foreign environment that's not, you know, the typical home environment, having a lot of leads plays, having respiratory activity recorded all through the night. One of the advantages of this, though, is that you do get a lot of information about sleep and a lot of the information about the brain activity, the different stages of sleep that an individual has. And I'll show a little bit of data that we've collected in that regard. The cons of this definitely requires a lot of tolerance, having these leads placed, being in this foreign environment. And this is, of course, is a very expensive approach, especially, you know, from a kind of a clinical research perspective. All right, so questionnaires. We published, again, a study, I guess now a couple of years ago, using the CSHQ. As I mentioned, it involved a research coordinator, either talking in person or by phone, giving the questionnaire, and involved something around 35 questions or so. So what did we find? So we found that kids with SYNGAP1 mutations have significant sleep issues. So on this scale, looking at the total score, any score above 41 is considered to indicate significant sleep abnormalities, and nearly all of the kids with SYNGAP1 mutations had significant sleep abnormalities. We again compared to kids with Phelan-McDermid syndrome as well as healthy control siblings, and not surprisingly, our SYNGAP1 kids were much more likely to have abnormalities in sleep than healthy controls. Interestingly, for this study, we found that kids with SYNGAP1 mutations tend to have more severe scores on the CSHQ than kids with Phelan-McDermid syndrome, and I'll get into a bit more detail about that in a minute. Again, with this questionnaire, we can break down the sleep abnormalities into different domains, such as bedtime resistance, sleep anxiety, sleep onset delay, and night awakenings. Again, found that for all of these domains, kids with SYNGAP1 mutations have significant issues. But again, interestingly, for many of these, we saw a difference between kids with SYNGAP1 mutations and kids with Phelan-McDermid syndrome. In general, for most of the domains, kids with SYNGAP1 mutations seem to have more severe sleep issues than those with Phelan-McDermid syndrome. And again, just kind of rounding that out, looking at parasomnias, daytime sleeping, sleepiness, sleep disordered breathing, which you can sort of think of as sleep apnea, and sleep duration, again, significant abnormalities in our kids with SYNGAP1 mutations. So why do kids with SYNGAP1 mutations have more issues with sleep than kids with a similar disorder, Phelan-McDermid syndrome, who I see these kids clinically, and parents very frequently come to me with sleep issues. So we looked at this in a bit more detail, and again, looking at whether age was a factor. And it turns out it looks like age is a factor, more so for Phelan-McDermid syndrome than for SYNGAP1, in that kids looking at total score of kids under 11 years of age, we saw a significant abnormality still in the kids with SYNGAP1 mutations, but did not see a significant difference between healthy controls and kids with Phelan-McDermid syndrome in this young age group. And this is in contrast to the older kids, 11 years and older, where we see significant abnormalities both in SYNGAP1, kids with SYNGAP1 mutations and kids with Phelan-McDermid syndrome. And we saw some other differences overall in some of the different sleep domains, such as sleep onset delay, where at least statistically, we saw no significant difference in the SYNGAP1 kids or the Phelan-McDermid syndromes with the healthy controls. But then when we looked at the kids 11 years and older, so this is again around puberty or so, both the kids with SYNGAP1 mutations and with Phelan-McDermid syndrome had significant issues with sleep onset. All right, so I'm going to turn now to the second study that we embarked on, which is to use actigraphy to get a little bit more objective and again quantitative data regarding sleep. So again, looking at this middle panel here, this involved kids wearing a smartwatch that records really movement. So these smartwatches have have an accelerometer so that we can record movement. So when kids are awake and active, obviously we see a lot of movement. When kids are asleep, we see much less movement, and that's how the algorithm that looks at this data breaks down whether someone's awake or asleep. And so the type of data that we get is shown here on the right, where you see all these squiggly lines, yellow lines, blue lines, black lines. This is the time of day where there's a lot of activity and the light's on, and the kid's awake. As compare that to sort of the teal blocked area, which is where the computer algorithm is saying that the kid is asleep, and that's because there's a little activity. Usually the lights are out during this period. So you can see it can differentiate those two, wakefulness and sleep. And so really early on, it looked like there was going to be something really, really interesting and different between SYNGAP1 kids and sibling healthy controls based on actigraphy. And that's really sort of illustrated here, where if you look at the time this child is asleep during a 1 week period on the left, SYNGAP1 patient, you can see the amount of time that they're asleep really varies dramatically from day to day. There's a lot of variability. And this is in contrast to the healthy sibling, where you can see, while there is some variability, it doesn't seem to be nearly as much. And so for this pair of patients, if you look at the quantitative data, and I'll have you focus kind of on that middle column there with total sleep time, you can see that if you look at the average actually is not much different between the SYNGAP1 patient and the healthy control sibling. But then the minimum amount of time asleep in the SYNGAP1 patient compared to the healthy sibling is significantly different, as is the maximum amount of sleep. So at least for this pair, there is a big difference between minimum amount of time and maximum amount of time asleep during during the 2 week reporting period, whereas the average ended up being exactly the same. So this looked really promising at first. But then we collected data from a fairly good number of kids with SYNGAP1 mutations and healthy controls. We took a lot of pains to make sure, I'm not going to show this, but to really age match and match based on biological sex so that those weren't variables that were very different and could skew the data. And so when we were able to collect enough individuals, we analyzed the data. And what we found was actually kind of disappointing, and that is that looking at all of the different variables, total time in bed, total time asleep, the latency from time to go to bed till sleep initiated, sleep efficiency, wakefulness after sleep, and number of awakenings, none of these were significantly different between our SYNGAP1 kids and healthy controls. And this was really surprising because for all of these kids, we also had data from the sleep habits questionnaire. And what we found is looking at the correlating data from the CSHQ, we saw in these same kids very significant differences in things like sleep duration and bedtime resistance, et cetera. So I did mention that, one of the factors here might be that some of these variables, there might not be a difference in the mean amount of time, for example, that they're asleep, but the variability could be greater. And that does turn out to be the case. We do see a greater amount of variability in our kids with SYNGAP1 mutations compared to healthy controls. And, in many of the variables that we looked at. But then, overall, we really did not see a statistically significant difference between the kids with SYNGAP1 mutations and healthy controls. So, again, why is this? Like I said, some of it could be that while there's more variability, that, in the end, the sleep need is the same. I tend to think that's probably not entirely true because, definitely what, you, the parents, tell me about your kid's sleep would suggest otherwise. And then, the data we collected from the CSHQ would also argue otherwise. So, we're still trying to work through why this might be. I think part of it may just be the technology, just is not great for our kids with neurodevelopmental disorders. This system has been used in other neurodevelopmental disorders, and A lot of that data has never been published because people find the same thing, which is that there's not a lot of robust data with the actigraphy or robust differences with the actigraphy compared to various neurodevelopmental disorders and healthy controls. So we're still sifting through the data a bit, but overall, that's kind of the first line of findings that we had from this. So as I mentioned before, the third approach to looking at sleep in kids with neurodevelopmental disorders is sleep studies or polysomnography. Again, this involves going to a sleep lab, usually occasionally can be done at home, and having kids have electrodes placed, a few electrodes placed on the head, as well as some respiratory monitors. Again, can potentially be very difficult for our kids with SYNGAP1 mutations to tolerate this, but we can get very good data, not just some of the parameters I talked about before, sleeping, waking, but also look at more detail at the different stages of sleep in kids with neurodevelopmental disorders compared to healthy individuals. And for this, we really just have some very preliminary data, but already emerging are some interesting findings. I'm not going to go through all of this, but these are all kids with SYNGAP1 mutations who had sleep studies for clinical reasons. So, that's one caveat to this is all of these kids are known to have sleep issues. And so that's the reason they were referred for a sleep study. But nevertheless, when looking at this data, a couple of things popped out. One is that it seems that sleep efficiency can be significantly reduced in these kids. So what is sleep efficiency? That's basically once kids go to bed, how much of the time that they're spending in bed are they spending asleep? And for neurotypical kids, we expect this to be around 85, 90%. And we saw for a number of kids where we had sleep study reports, significantly reduced sleep efficiency, really in the 60% range, which is very, very low compared to what we expect. And then the other thing that I found to be very interesting is the percentage of time that's spent in REM sleep. So REM sleep is a part of sleep that's really important, especially for learning and memory. It's been found that having a healthy amount of REM sleep is important for any individual to develop new memories. And normal for kids or adults is somewhere around 20 to 25% of the time that they're asleep, they spend in this REM state. What we saw in the small number of kids with SYNGAP1 mutations, that a lot of them have a significant reduction in time they spend in REM sleep. In fact, some of them had very, very little REM sleep, down to like 2 to 4%. The reason for this is not clear, although I will say in other disorders, there is some correlation between epilepsy and reduction in REM sleep, which may play a role here. But what we can definitely say is that, with a significant reduction, this could lead to impairments in ability of these kids, contribute to ability of these kids to have difficulty developing new memories and learning. All right, so that's really all of the new data that I'm going to show today. And I have just about maybe 10 minutes left. So I'm going to just rapidly go through some of the studies that we published before. Again, this is mostly just as a reminder, but also for any new families that have not heard me talk before. So one of the earliest studies we did, published in 2019, was looking at development in kids with SYNGAP1 mutations. And what we found from this early study is that kids with SYNGAP1 mutations really have issues with developmental progression, really in all domains of development. So shown here are some key milestones that kids go through during development, including first words spoken, time sitting unaided, time walking unaided, scribbling, and using utensils. And for all of these milestones, which represent gross motor, fine motor, language development, we saw significant delays. Looking at this a bit more quantitatively, our kids with SYNGAP1 mutations have delays that persist through development in gross motor acquisition of skills. They do continue to learn, continue to develop, which is why I always encourage all of my families, as much as possible, as much as they can tolerate, as much as insurance will allow, to continue in physical therapy, occupational therapy, speech therapy, all of these therapies, through much of the, certainly early development, but really potentially through much of the lifetime, can help with acquiring new skills. And although our kids with SYNGAP1 mutations, again, acquire these skills much more slowly than neurotypical kids, they do continue to acquire these skills. And this is true, again, for gross motor development, fine motor development, as well as language development. Looking specifically at language development, we do see, probably for this developmental domain more than any other, quite a bit of scatter, at least in the early developmental stages, where you can see on average, like at 50 months of age, these kids have skills at around 20 months of age. But then you see a lot of scatter where some kids are nearly at a neurotypical level. And one question that we've wondered about is why is this? And there could be a number of reasons, but one could be that there's something about the type of mutation or where a mutation is in the SYNGAP1 gene and its correlation with developmental progression. And we potentially see something interesting in the kids that we identified have a bit higher developmental acquisition in language. And that is, some of them have either missense mutations, which may not fully render the mutated allele completely deficient, or we have mutations very early in the SYNGAP1 gene, such as this kid with the mutation at position 52, which probably only affects a subset of isoforms of the protein. So kids with, so at least some rare kids with mutations like this might have slightly different developmental progressions than those kids with the more classic complete loss of function mutations kind of in the middle of the gene. We also looked at seizures in some detail, and I think most in this group kind of appreciate that it seems like the most common seizure type are these atypical absence, or frequently referred to as absence with eyelid myoclonia. But we see a wide variety of different types of seizures, including more classic tonic-clonic, generalized tonic-clonic seizures, or you might have heard them referred to as grand mal seizures. We also see some focal seizures. Atonic seizures appear to be relatively frequent. Myoclonic seizures, relatively frequent. This is a little bit of older data. I do have one child now with the SYNGAP1 mutation who did have infantile spasms, which are kind of really early, relatively severe seizures, but that seems to not be terribly common, at least in kids with SYNGAP1 mutations. We also looked at medications used in these kids, and at least retrospectively, whether they're efficacious, and found a few medicines that seem to be a little bit more efficacious than others, including valproic acid, lamotrigine, as well as clobazam. And these are some medicines I use fairly commonly in these kids. And then some medicines that really seem to not work very well at all. Either they don't improve seizures or they're not really well tolerated. And this includes what are called sodium channel inhibitors, such as oxcarbazepine, as well as cosamide and rufinamide. Perampanel is interesting. There are some sort of case studies published suggesting that maybe low-dose perampanel helps with not seizures, but some other aspects of SYNGAP1 phenotypes. At least for seizures, we've not had a lot of success with perampanel. And then we move on to EEG abnormalities. So probably most, if not all of your kids at some point have had an EEG. This involves placing electrodes on the scalp and then recording electrical activity. And shown here on the right is sort of the type of graph that neurophysiologists get and what they interpret. And looking, again, this was a few years ago now, but it's really held up over time. Kids with SYNGAP1 mutations, they tend to have what are called interictal abnormalities, most commonly in the occipital lobe. These interictal abnormalities are neurophysiologic abnormalities that are not associated with seizures, meaning the child is not having a clinical seizure at that time. but we can still see electrical abnormalities by EEG. And again, the most common type that we see is in the back of the head, which is the occipital region of the brain. Don't know why kids with SYNGAP1 have this pattern. This is not something we see in all neurodevelopmental disorders or even most neurodevelopmental disorders, but does seem to be fairly specific for SYNGAP1 kids. Just showing you a couple of EEGs here. This is an EEG from a kid that was having an atypical absence seizure with eyelid myoclonia, and you can see kind of on the left side of the graph, see a lot of really tight squiggly lines, and then suddenly, these really high amplitude, really broad waveforms that indicate an abnormality associated with the seizure. And then again, I'm not going to belabor this point, but we very frequently see abnormalities in the posterior part of the brain, the occipital part of the brain, even in children with SYNGAP1 that are not having a clinical seizure. We see these abnormalities. And then behavior abnormalities we see very frequently in these kids. I'm not going to go into a lot of detail about this, but the types of activity abnormalities that we see include hyperactivity, self-injurious behavior, aggressive behavior seem to be very, very common in these kids. And I work with a lot of the parents that bring their kids to me on this. Pain tolerance is, high pain tolerance is very common. And then many of these kids are on medications, and on the right here are some of the medicines that we commonly prescribe. I'll just briefly mention that it seems like, although these kids are hyperactive, those kids that I've seen where stimulant medications have been tried, these are like Focalin, Ritalin, these sorts of medicines, very rarely see much improvement and very often see a lot of worsening of behavior. So I tend to stay away from those and use other options. And then sensory processing, this is data from Gavin Rumbaugh's group showing that mice that are deficient for SYNGAP1 have a lot of sensory processing issues. And so we looked at this in kids with SYNGAP1 mutations. I'm not going to go into all of the details of this, but just suffice to say that we see a lot of abnormalities in sensory processing in our kids with SYNGAP1 mutations, and very comparable to those kids with Phelan-McDermid syndrome. And looking at kind of in detail at the types of sensory abnormalities that these kids have, can sort of broadly think of sensory abnormalities as being either kids that have a lot of seeking behaviors, that is, they seek out a lot of sensory input, or they tend to avoid sensory input. For Phelan-McDermid syndrome, we kind of see both. We have some kids that have both sensory seeking and sensory avoiding behaviors. We have some kids that just have avoiding behaviors, and some kids that just have seeking behaviors. Actually, quite a few kids that with Phelan-McDermid syndrome that just have seeking behaviors. This is in contrast to our SYNGAP1 kids, which are a bit different in that a lot of them have both seeking and avoiding behaviors, but very few of them have just seeking behaviors in a very small number. And then only a few have only avoiding behaviors. So they seem to have both types of sensory abnormalities, sensory seeking and sensory avoiding. And then very quickly, I'm going to talk about a gait study that we've had ongoing for a while now. We're using a number of really objective ways to analyze gait. Again, I've talked about this before, so I'm not going to go into a lot of detail because of time, but basically we can use what's called a motion capture system to look at joints and joint angles and stride length, stride time, etc. This does require kids to tolerate putting on this suit where we can attach these reflective monitors or reflectors at various joints and various parts of the body. Usually we restrict this to the lower extremities. Then we have kids walk either over a carpet, which can also measure some activity like stride length and time, as well as force of strides, or we have them walk on a treadmill so that we can use this motion capture system to look at gait abnormalities. And this is the kind of data that we can collect. So although there are cameras, they actually only record the reflective balls that are, again, placed on joints or on surfaces of extremities. You can see at the bottom there, that's sort of some of the raw data that we get. We've published one paper looking at one individual with a SYNGAP1 mutation compared to a neurotypical control, and did see some significant abnormalities that primarily involve differences in symmetry. Especially at the ankle, there should be a significant amount of symmetry between the left and right side, and we saw that in the neurotypical control, whereas our individual with SYNGAP1 mutation had a high degree of asymmetry, which ends up, other than causing a lot of instability, also leads to a greater amount of energy expenditure because of having to rebalance with so much with each step. And so we're now, we've expanded, we've recorded data from 8 individuals with SYNGAP1 mutations, hoping to record from 2 more to kind of collect a preliminary data set to analyze and be able to say something a bit more broadly about gait issues in kids with SYNGAP1 mutations. So to just summarize what I've talked about today, I just briefly discuss what SYNGAP1 is, and that mutations in SYNGAP1 are one of the most common monogenic causes of autism. But kids with SYNGAP1 mutations don't just have autism. They have really a complex neurodevelopmental disorder associated with sleep disturbances, abnormalities in socialization, really delay in all developmental domains, also very commonly with seizures, characteristic EEG abnormalities, also have sensory processing abnormalities, as well as gait abnormalities that we're looking at now in more detail. So a number of people to thank, a lot of people that I've worked with here at Texas Children's and Baylor College of Medicine. We work with the University of Houston and Charles Lane there for the gait study. Thanking my collaborators, Connie Smith-Hicks and Andy Stanfield, who've been really key for the sleep study that I talked about. And then, of course, all of you who've participated in our work and helped us get the work done through various mechanisms like funding, et cetera. I really appreciate all of you and working with me. Our goal is to, with all of this work, is to develop those endpoints that can be used for future clinical trials. And then talk about the work that we're doing in the lab related to, molecular understanding of how mutations in SYNGAP1 affect brain activity or potential mechanisms for treatment, which we're working on. And a number of really nice papers have come out recently with some potentially new approaches. So I think there are good things on the horizon for SYNGAP1, and hopefully some of the work that we've done here can aid us in developing those clinical trials in the future. So thank you very much. I guess I'll take questions if there are any.
Yeah. Thank you, Dr. Holder. We're going to hand it over to Catherine.
Thank you so much, Dr. Holder. What a tour de force. That was a lot of different, a lot of data in different domains. So thank you for that. I want to encourage people in the webinar to either put your questions in the Q&A. And pardon me, I'll just read off the first one here, or there's only one right now in the Q&A. I have a lot of questions, but since I can go on, I'm going to ask what Erin's question is first. When you're looking at the sleep, could differences, things being different than you expect, could that be impacted by supplements like melatonin and stuff? Because our kids have such a hard time sleeping that we do everything we can to try to get them to sleep.
Yeah, excellent question. And absolutely. We did, record whether sleep supplements or medications to help with sleep were being taken. But what I would say is, even with that, with the CSHQ, a lot of those kids were taking the supplements and still had a lot of sleep issues. So we still see that, whereas in actigraphy, we did not see that. And again, same kids using the same 2 instruments saw different results. So, I think it's a good question and very on point that can absolutely impact, any study looking at sleep. But, specifically for, those 2 approaches to looking at sleep, I think there's a little bit more going on, and probably just, the technology that we use is not good enough to really capture those symptoms.
Yeah, while you're talking about that, I want to ask, with the actigraphy, was there any ability to look at heart rate?
Yeah, no. Although I said they kind of look like smartwatches, actually, they kind of look like dumb smartwatches.
Yeah, but like my Fitbit tells me, like I have a Fitbit, right? And
Yeah.
so that gives me some information. I think it's based mostly on heart rate.
Yeah. These are really old devices. I mean, they were new when we bought them, but they were developed a long time ago, and so
Right,
they
right.
really can only measure activity and light. They do measure, whether light's on or off, but they did not measure heart rate.
Oh, interesting. They measure light. Okay. That's not what I would have thought of. Okay, well, Megan in the chat says, are there any strategies to help with the sleep?
Sure. So strategies, I guess, that I use kind of clinically is what she's asking. So as you said, I also try everything. Certainly, to begin with, I try melatonin, which rarely alone really helps kids with SYNGAP. Occasionally, the younger kids will respond well enough to melatonin. But then, after that, there are some other classes of medicines. So things like clonidine can sometimes help, especially with sleep initiation. And then some of the kids that I see clinically use trazodone, which is an SSRI that, developed initially as for depression. Doesn't actually work well at all for depression, but has the side effect of making people really sleepy sometimes. So I try that as well. But as you said, I mean, some of these kids, will respond great to those medicines, and some don't respond well at all. So, but those are things I try. Of course, I also encourage parents, keep lights, turn the lights out, an hour before you want your kids to go to sleep. Don't let them have devices and things like that. But these kids really have pretty severe sleep issues sometimes.
Yeah, I just want to put my own 2 cents in there. I don't know how old Megan's child is, but when my child's 22 now, and he sleeps much better than before. But when he was young, like he wouldn't nap. Like as a newborn, he wouldn't really nap. He'd barely sleep. It was really strange, and he didn't even change his day-night cycle like most newborns do. It was months before he even really slept at night. And I think that was a sensory thing. Like I think he was kind of shut down during the day, but then he'd be awake at night because there was less going on. I think he could handle it better. But I just wanted to say that early on he would. I've read and heard that toddlers they get nervous when they start feeling sleepy, and so they start kind of running around to wake themselves up. And so what I wouldn't say this worked for me, but what I did was I would just sit next to his bed and just keep telling him to lay down. And so like every 10 to 90 seconds, he'd start lifting his head, and I'd say lay down, and he'd put his head down again. We could do that for an hour or two. I mean, it's a long kind of boring time, but it's interesting to try to not be interacting, but also telling him to lay down, and he would. And then eventually he'd get tired and he'd start kind of like rubbing his hands, and then he'd go to sleep. Another thing was that he really had to have a light on. And so I took all the lights out of his room, and I put a 20-watt bulb in the ceiling lamp. And so I kind of like reduced the amount of light. So he had very little light in his room because he would jump up from bed and turn that on. Like he had to have that on. So I just want to say that anytime you have something like that is to kind of recreate the environment so that the thing they are trying to change is already changed for them. So that's my advice.
No,
Okay, so
great ideas.
let's see. With sleep, I also wanted to ask, I wonder if there's a way to have like a mattress pad with a sensor in it that just sort of shows movement. And it wouldn't just show movement, it would show like how much of the bed they were moving on, right? Because I wonder if like distance moved would be and then like not in bed at all, right? Like the sensors can tell you that. So I wonder if that is something that would help. And then, let me see, what was my.
Yeah, so I'll just answer that. There are instruments that have now been developed that do exactly what you're saying, that you place them under the sheet. So instead of, having something on your wrist measuring activity, it's just in bed, how much time is spent moving in bed versus not, and calculate sleep from that. So we did not use those.
Yeah.
That might be good.
Yeah, I'm just thinking, because I don't think the actigraphy is ever gonna get good enough with our kids' sensory, unless you had like an entire sleeve. Like if you had the entire, like putting a watch on is never ever going to work for my child. Like he can't put a bracelet on for being in the hospital, right? Like they have to like pin it to his clothes on the back, because he can't do it. And so it's like but I think that what that means is that feeling this part being different from this part is not okay, right? And so if it was a whole sleeve that had some sort of, like if you're using compression garments and have like sort of a whole feel, so you're not really feeling the difference, I think that would be a way to work it out. Although that's not the same as just going to Best Buy and buying a smartwatch or buying a Fitbit or something.
Right.
Okay, let's go to Marta, Dr. Marta DeHied. Marta, do you want to get on the call, or did you want to be? Well, actually, I don't know how to unmute you, so I'll just say it. She says, thank you so much for the great presentation. Based on your clinical experience, what therapy do you find more valuable for behaviors and autism in SYNGAP patients?
Yeah, great question. So, certainly one of the first line therapies we recommend is ABA therapy for our kids with autism and SYNGAP1. I will say, this is a challenge I'm sure many people here have had. I talked during my presentation that only about half or so of kids with SYNGAP1 mutations are actually clinically diagnosed with autism. I think that's probably a bit of an underestimate, but there are definitely kids with SYNGAP1 that do not manifest all of the phenotypes necessary for an autism diagnosis. That doesn't mean ABA therapy, excuse me, would not be beneficial. Apologize. ABA therapy was actually developed for kids with intellectual disability at Kennedy Krieger. It's not developed initially for autism. And it was demonstrated to work or to overall lead to improvements in kids with intellectual disability. However, it's only generally approved by insurance companies for kids with autism. Anyway, that's just a bit of an aside, but that's always my first line recommendation. And then in terms of medications, I can give you a list of medicines that we use. I cannot tell you, oh, this is the medicine for SYNGAP, and these kids always respond to this medicine or that medicine. But there are certain ones that we try. Certainly, I can talk about risperidone in general is the only FDA or one of the only FDA approved medications for behavior in autism, not for SYNGAP specifically, but for autism. And some of the kids that I see use risperidone and have some improvement. I've had some that it didn't work or the side effects were intolerable. Like the antipsychotic medications and then the alpha-2 adrenergic agonists are another class. So clonidine and guanfacine are some medicines that I try. Like I said, with some improvement in some kids, but some kids don't respond.
Great, thank you. When you talk about therapies, you mentioned in your presentation that patients with SYNGAP are continuing to learn and are, getting skills over time, and that continuing with the therapies is important. this is something that I ran up against, of course, having, you know, my son's 22 now, he did all the therapies early on. And after a time, they start saying, okay, well, insurance will pay for 8, but you have to show that he's improved over 8 sessions, or we're not going to keep paying, because, and it's really a burden, you know, these are set up for the privilege of the kids that have the brain with the plasticity that can actually do this in a shorter amount of time, right? And so our kids have this larger obstacle. They can learn, but it's going to be more on their own time scale. It's going to be harder to see over 8 sessions.
Absolutely.
I'm wondering if there's any way you can write a case study or a opinion piece or something that is published, so that we can hand this to our insurance company and start advocating with a publication.
Oh, that's a great point. I hadn't thought about that, but
It's all we think about.
well, certainly the data in terms of development continuing over time, you know, we published that in 2019 from the very first paper we published on SYNGAP. But it's a great point that you know, one, that may not be enough for insurance companies to say, oh, okay, we understand, we'll approve it. So yeah, it's a good point, and it's definitely something I've run into as well. And I've certainly, you know, one thing I've done is written, you know, appeals to try to, you know, get that reversed with very variable success. Depending
Absolutely. But as a you know, since I don't want everyone to go to you to write every appeal, it would be awesome if you wrote one paper that just said in very plain terms, this is what's happening, this is their obstacle, this is part of the disease. If you're going to get a free, and you know, you could put in verbiage that includes school, you know, free and appropriate education. You could include verbiage that would be you know, recognized by insurance. I'm not saying insurance would then pay, of course. Like, I think the whole job of the insurance company is to try to not pay for things. Absolutely. And then you have to like get your ammunition in order and like try to shoot through the shield so that you can actually get payment. Yeah. So I understand that, but it would be so helpful if you or anyone else, like, even it'd be more helpful for each clinician to do this separately, so that we had multiple individual articles that could show, rather than like a group doing it together. Right. That's my own personal opinion. I don't know if that's actually what would happen. Okay, I'll stop talking about that, but that's got me very excited. Okay, so Olga asks, what age groups were involved in the GATE study, and is surgery ever warranted?
So for the GATE study, I mean, in general, we're looking at individuals that are under 18, and I think down to 4 years of age. We didn't want to go much below that just because it's difficult for younger kids to sort of participate, and their gate's still developing. So, you know, we're really looking at, after gate should be, you know, more or less developed, you know, what are the differences from neurotypical. So basically like 4 to 18. Oh, is surgery, I'm not sure what she means by surgery warranted.
I think sometimes people have, oh, I guess Olga can ask you herself. Go ahead, Olga.
Hi, it's all right. Thank you for, yeah, well, Drew's already, he's had, speaking personally, obviously, Drew's had surgery, and we just consulted again with physical medicine, and there are other surgeries that would help repair everything. I mean, to where it should be as far as rotating the bones, etc. So he would walk in a more normal manner. And I just didn't know if how many kids, if they've done the GATE study, have had any surgery, or these are just typical, just kids with maybe using orthotics and having physical therapy. Okay. I mean, I thought physical therapy was one of my first targets when he was younger. And once he started walking, everything seemed fine. And then about 8 or 9 years old, things just went awry. And we were fortunate to have some surgery before his growth plate we were able to do a couple of procedures to help, but he's still got issues.
Got it. No, okay, I understand now. What I can say is that in general, among the kids that I see with SYNGAP1 mutations, uh, qualitatively, many, if not most, have gait abnormalities. I've had very few that have needed surgery, like exceedingly few. So all I can say is it's not common that the gait abnormalities lead to surgery. So yeah.
Okay.
Olga, I have a follow-up question for you.
Yeah, sure.
Is that due to hypermobility? Is that like tendons and ligaments needing to be shortened, or is that something else?
Yeah.
Is that orthopedic? Is it more bones? What's
It's all
happening there?
orthopedic. Yeah, it's orthopedic. I mean, there are, if he could tolerate the next round of surgeries, which would be rotations and things like that, but he's not developmentally at a place cognitively where we, is he going to sit for 6 weeks in casts? That's kind of what we would, but he said this is something that could be done when he's 20 or 30 if we want it. So we're just kind of monitoring the situation right now and still attempting to use orthotics.
Yeah, I just would say for us that seeing my son at 22, he'd be way better able to handle that at 22 than at than at 15, 16, 17.
Yeah. Okay. So that's
and then Dr. Holder, I wanted to ask you also about gait. My son walked at around, I don't know, 18 months, and he definitely had a little bit of a different gait. He's had that the whole time. He didn't grow for quite a long time. So he had 4 years, like when he was 14 and his bone age was delayed by 4 years. So we see this with a number of Syngapians where they won't eat, or they're eating, but they're not gaining weight, he kind of like drops off his growth curve for a while. He then was able to restart, and he grew pretty fast. And then his gait got much worse. So I've always wondered, I did a lot to help him figure out his body map, right? Like I did all this baby massage. I was always giving him like input in his body because he had such a high pain tolerance that I figured he wouldn't know where his hands were. He'd want to eat and he'd go like this. And if I touched his hand, he'd reach out and grab it, right? So he didn't have you could tell he wanted something, but he didn't have an awareness of where his body was and what was possible for him. So I did a lot to give him sensory input to give him, because in our brain, our body map takes up the whole brain, right? So, so I was wondering if after his growth spurt, his lack of synaptic plasticity didn't really allow him to change his body map. And so he's like moving with these long levers and his body map is still for a little guy. I was wondering if that's partly why he has such a much harder time moving around and doing things now.
Yeah, it's a great question. I mean, I don't have the answer to that. It's certainly plausible. It's hard to say, but
yeah, he used to be able to run, and I wouldn't say he ran in a really typical way. Like he looked different than my other kids who could run and sprint, but I don't think he ever really, like now he kind of lopes. He's got like these big heavy feet, and he's got, he doesn't really run, which is, good, because I probably couldn't keep up with him. He's taller than me now.
Right.
Okay, so let's go to Megan says she's 2 and will wake up multiple times a night. That's probably because I asked. I don't think that's a question. I think that's me just asking her about her. So Marta said, what about seizure control and its relationship with decreased behaviors and improvement of sleep?
So that's an amazing question, and one that we've not addressed specifically, but should be, and not just about seizure control, but also about degree of EEG abnormalities, and does that correlate in any way with behaviors, with development? And we don't know the answer to that, certainly for SYNGAP1. I've been interested in this for a while for Phelan-McDermid syndrome. I've looked into the data that's available for autism in general. And, it's for all of that, it's still inconclusive, I would say. You would think that in general, if someone is having a lot of even interictal activity, increased activity, that could impact their development. And you would, hypothesize that if you gave medication to try to reduce that interictal activity, maybe that would improve development. The data just doesn't exist for that, as far as I know, unless something has come out really recently. People have tried this in very small numbers for autism. There are only some seizure medications that are known to reduce this, what I keep calling interictal activity. So these are abnormal discharges, but not associated with a clear clinical seizure. And in those very small studies, there was no significant improvement with or without medication. Again, these are really small, and
when you say improvement, do you mean improvement on cognition? What's the improvement they're looking for?
Yeah, so I'm stretching a little bit. It's been a while since I read these. I mean, they used instruments. I feel like one of them used what's called the ABC, the Aberrant Behavior Checklist. So looking kind of broadly at behaviors. I'd have to review those, but there were endpoints that are kind of standardized endpoints.
Yeah.
And I should say, I said that there's no convincing evidence. Some of those studies do show improvement, and then some don't. And they're all generally open label, so everyone knows that everyone's getting the medicine. So that's a confounder as well. So what I'm saying overall is, although I think the hypothesis is very interesting, it just hasn't been tested, as far as I know, for any neurodevelopmental disorder, and certainly not for SYNGAP1. But a very interesting question.
Can I add an anecdote?
Mm-hmm. Absolutely.
My son has had the absent seizures his whole life. He's had the eye rolls, but he was born in 2001, and I was told repeatedly when he had EEGs that just had epileptiform discharge activity, but not actual seizures, that he wasn't having seizures. So we weren't on meds at all, and he wasn't diagnosed until 16. So he started having drop seizures at 18. So that's when we started on various medicines, and now he's on valproic acid and Onfi. But when I first started the valproic acid, he went from being able, like, if I asked him a simple question, like, give me your cup. That's not a question. That's a command. I'd say, give me your cup. I would need to I'd need to repeat it several times. I'd need to give him prompts. I'd need to show him, point to where the cup is. Then when he had it in his hand, I'd need to put my hand near him so he could put it to my hand, right? Like it was a scaffold. He could do it, but it was very scaffolded with lots of input for me. After a while, I remember saying to him, give me your cup. He looked for it. He got it. He looked at me. He handed it to me. I was like, you're kidding me. So he did the same thing, but it was without all these supports, right? Now, he didn't start talking. He didn't start solving puzzles. He didn't start, he didn't do more things. But to me, that was a very significant change, but it also had its limit, right? There was an improvement, but it was very localized and it was limited. It didn't just say, okay, now he's going to develop the way he's supposed to develop. So that's my anecdote.
Yeah, and I've definitely seen that frequently in clinic. I can't necessarily say for SYNGAP1, for some of the patients probably, but for neurodevelopmental disorders in general. I've seen that anecdotally, but especially if there's a high seizure burden, that getting some improvement can lead to better ability to learn.
Mm-hmm. Yeah. So when somebody was asking, or when you were discussing the medicines for behaviors, I noticed you didn't say something like fluoxetine, so something that's like an anti-anxiety. That's been I know that's helpful for some kids. It's been incredibly helpful for my kid. And through school, whenever there'd be a week with really increased behaviors, we would up the fluoxetine and they'd come back down. So that we sort of stepped that up, and that's been helpful for us. Yeah. I just wanted to say that leading into Marta's next question. What's your theory as to why SYNGAP has such marked aggression, not explained by ID or autism itself, and appears much worse than other similar diseases?
Yeah, so great question. And that's just an oversight on my part. I have a few kids that take SSRIs with SYNGAP1. So sorry, that was just an oversight for me. Um, Marta asks all the great questions that I don't have answers to. I think she knows I don't have answers. But it's a very important question. Yeah, I mean, there are other disorders, and I'll, somewhat use Phelan-McDermid syndrome as an example, since that's, the other population I see a lot. It does seem that in general, kids with SYNGAP have more aggressive behaviors. It's more broad in the SYNGAP1 kids. Now, some of the Phelan-McDermid syndrome kids definitely have aggressive behaviors, and some of the SYNGAP1 kids don't have very many aggressive behaviors. But I would say in general, anecdotally, of course, again, no data to show you, the SYNGAP1 kids seem to have more aggression. And why is that? I mean, I can hand wave and come up with all sorts of hypotheses. I don't have, any data. SYNGAP1 in general, deficiency of that causes greater, hyperexcitability of neurons, whereas Phelan-McDermid syndrome, that tends to decrease excitability of neurons. Maybe that somehow plays a role, but maybe the expression pattern of SYNGAP1 compared to the expression pattern of SHANK3 in the brain could have some impact on that, but that's all just, hand waving, and it's a very important question that we don't know the answer to. At least I don't.
Thank you, Dr. Holder. Okay, so our next question is from Nancy Kessler. She's a very beloved member of the SYNGAP community, and she's asking, she's the sister of Karen, the oldest SYNGAP patient that we know. And have there been studies looking at gait decline with age? And she says, thank you for this excellent presentation and study.
Well, first, thank you so much for listening to me. No, so that's a really interesting question as well. There have not been any studies looking at longitudinally at gait. Actually, there's not been much in terms of longitudinal studies for SYNGAP1 at all, which is really an important next step. Everything I showed you today was cross-sectional. Meaning just, one patient, one point in time, several of those. But looking at gait longitudinally, also looking at development and sensory processing and sleep, I think these would all be great to look longitudinally. And I will say, just as a little bit of a plug, we are looking longitudinally at sleep using the CSHQ. Those of you that have participated, many of you probably got a second round of calls to collect data for a second year a while ago, and we should be collecting a third year soon. So hopefully that will provide some information at least about sleep. But I agree, all of the phenotypes should be looked longitudinally.
Are you still using? Is your gait mat still in use? Is it still set up, and can you still? Can you get the patients that were in the first thing? And I can imagine even like every 2 years or 3,
Yeah.
Not needing a lot of granular data points, but maybe even a longer study.
Yeah, so again, another excellent point. And I've talked to Dr. Lane, who is our, so I didn't go into detail about him. He runs Center for Human Performance at University of Houston. So all he studied all of his life is gait in various populations. So he started, I think, with astronauts. Looking before and after spaceflight. And he's also looked at like Parkinson's disease, some adult disorders, and really in kind of the last 5 years or so, I started working with us at Texas Children's looking at various neurodevelopmental disorders, first publishing in Rett syndrome. And then he's worked with me for SYNGAP1 and some other neurodevelopmental disorders. So yes, the gait lab is still up and operational, and it would be really interesting to look longitudinally with the same kids.
Okay, and then Marta has I think more of a comment. She says prophylactic anti-seizure med there's a study on tubular sclerosis. I'm sorry, I'm not sure how to, I know that disease, but I'm not sure how to say it, with clobazam showing cognitive improvement. So I guess there is a little bit.
Yeah, and that's a great. Thank you again, Marta. I do now recall that study. That's a fairly recent study looking at clobazam. So yeah, thank you so much.
So, and then finally, with your gait suit, I was wondering if, I was just thinking all that gait stuff could be somehow transitioned to the bed, right? So you could have, instead of a gait mat, you could have a gait mattress pad, and then, you know, like not gait, but a pressure mattress pad, and then a suit that has like little points on it. So maybe, you know, like sleeper jammies that have something, and then low light in the room or infrared. I don't know what it would be, but I know there's a lot of technology these days that.
Yeah, and there is a lot of new technology coming out. One thing I'm really excited about is there is new technology that's being developed to actually record PSG's polysomnography sleep studies at the home using less
Fewer leads or.
Yeah, it's like you know, something that can be just placed on the forehead. Again, this, you know, could be an issue in terms of tolerance for our kids with SYNGAP1 and all of their sensory issues, but doesn't have all of the leads going everywhere. You know, it's uses Bluetooth, I guess, or something to transmit the data. So, you know, a lot of new technology, I think, that can maybe address some of these questions.
Yeah, I wonder if there'd be any info in blood pressure and heart rate. Like I feel that our kids have, like when I think about my young children sleeping, I feel like their blood pressure and heart rate was different while they were sleeping, right? Even if one moved, and even if they both moved. And I'm also thinking that the REM sleep, that like for the Fitbit kind of algorithms for REM sleep, that those might not be since they're made for people with that do go into REM sleep, that you might look asleep, you might look like you're in REM sleep in the actigraphy, but actually be awake, right? Like you might not, like the algorithm might not be catching what's happening for you. Sure. I'm just wondering if the actigraphy is kind of just off somehow, but you're saying it's only movement and.
Yeah. So the actigraphy actually can't distinguish any of the phases of sleep. It can only. The one we used anyway. Only
yeah, so it's really it's interesting. It's puzzling, right? It's puzzling what that difference was about.
Yeah.
Okay, well, do you have any last words for us as parents?
No, you know, I just want to thank everyone who's participated in all of our studies. You know, you guys have been wonderful, and I really appreciate it. And you know, I would say, for neurodevelopmental disorders in general, but for SYNGAP1 in particular, I'm more optimistic today than I was, you know, 5 years ago when I started, that we're on the right path to clinical trials and hopefully treatments in the future.
Yeah, thank you so much. It's been such a pleasure to see your slides, hear your words, and you gave a really amazing introduction to SYNGAP1. I really appreciated that, and thank you for all your research, and I hope lots of patients continue to come see you.
All right, well, thank you, and everybody have a good day.
Okay, bye.
Thank you, everyone.